This video is actually going to be pretty short because it's talking about the energetics of transport, and that can be a little bit complicated and involves some math. So not much for here. And then we'll see some examples of transport proteins in the third lecture for chapter 11. But before we get into that, just a brief side note about membrane fusion. So one of the other things that membranes can do is fuse, and the reason that this can happen is because they are not polymers. So this group of lipids is associated into a membrane, but they're not covalently linked, right? Only each little baby one, that's a covalent molecule. So one of the cool things about them is they can grow. You can add extra lipids to a membrane and it can expand, and two different membranes can fuse. So what you have to do in order to get membranes to fuse is you have to first get them close. And this is a bit of a tricky part actually, because if you'll recall, this is a polar section and this is a polar section, and in many membranes actually they're both negative. So we don't really like to bring two negative things together, so that can be a little bit tricky. This is usually really the hardest part. But if you can get them close enough, then they don't want to be bumping up against each other. So instead what will happen is the inner parts will start to join together so that we don't have these negative things bumping up against each other. And eventually you will get just sort of an insertion of that guy. So this will sort of break, this will break, kind of just insert it in. So membrane fusion is important in all sorts of things. It's any sort of movement of a vesicle throughout the cell. So if you want to move something from here to there, you might have to bud off a vesicle, and then you might have to fuse it again into another membrane. So membranes are going to fuse or separate for a lot of different reasons in cells. So one is to deliver things from the inside of the cell to the outside. So you can bud vesicles off of things like the Golgi complex, and then if you fuse this vesicle with the plasma membrane, then you can release what's inside. So the budding itself would actually be, you have to fuse around the end, right? After you sort of bud it off, then this bit has to fuse. Then the exocytosis, there's another fusion. Endocytosis, which we've talked briefly about, the caveoli, that requires fusion of this bit of the membrane with this bit of the membrane, and may require fusion later on as well. Say fusing the endosome with a lysosome or something like that to maybe degrade what's in there. This also can happen between different types of organisms or different types of cells. So one situation is viral infection. So in order for a viral particle to infect a cell, the virus first, which has a membrane around it, is first going to have to fuse its membrane with the cell membrane, and then it can essentially inject its viral material into the cell. So that's probably something we generally don't want happening, right? But then something that is important that it'd be able to happen for any of us to be here is the fusion of the sperm and the egg. So, right, so when the sperm cell, in order for the sperm cell to get into the egg cell and mix up the genetic material, the sperm membrane has to fuse with the egg membrane. And there are plenty of other examples out there, so I just don't want to give you the impression that this is all of them. This is sort of a variety to give you an idea. So we're going to talk about one specific way, and this is actually used in neuronal signaling. So what's happening here is we have vesicles that are filled with neurotransmitters, which are just little molecules that transmit cell messages throughout your nerve cells. And these are inside the cell in secretory vesicles, and upon a certain signal, you're going to release them to the outside of the cell. So the fusion we're talking about is with the vesicle in the cytosol with the cytosolic side of the plasma membrane for an exocytosis. All right. So this is just showing you how this works, and other membrane fusions often happen in relatively similar ways, but with differently named proteins. So the proteins that are used here are called snares. And there are proteins both on the vesicle and on the membrane, and the vesicle ones are called V snares for vesicle The membrane ones are called T-snares, and I don't honestly remember what the T stands for in that moment. But these are proteins that when they're in contact with each other are going to form coiled coils and sort of wrap themselves around each other. And remember that proteins have a cooperative binding. So once you get these the ends of these together and they start forming their coiled coil, what's going to happen is they're going to want to keep folding up until the entire thing is a coiled coil. That is going to literally pull the vesicle close to the plasma membrane. So they're pretty long things, so they can reach out and they can grab each other when the vesicles are still a little bit far away. SNAP25 is just another protein that's involved in this whole thing. It also gets kind of wrapped up in there. And so so as it's wrapping up, it's going to pull this together, and eventually they're close enough that the outer leaflets start to fuse, and then the inner leaflets fuse, and then we create the pore. And then, so once this happens, then this has now become part of the plasma membrane, and other stuff can come along and sort of recycle these proteins. So now moving on to transport across the membrane. So what we're talking about here is moving usually an organic molecule, sometimes a non-organic molecule base, but not like a huge, large protein. We're not really talking about that. We're talking about smallish kind of things across the membrane. This can be either in or out of the cell or whatever. We're not going to differentiate at this point between that. And the reason we need this is because of this hydrophobic bit of the bilayer. Only a very few specific things can cross it on their own. So essentially the only things that can cross on their own are very small, very non-polar things. So something like oxygen, O2, because it is very small and also very nonpolar, it can actually just diffuse across the membrane. It's okay being in here. But most other things are not. Ions are going to be highly charged. That's not going to want to go into that nonpolar thing. Be like trying to mix up salt into oil, right? Doesn't work. And anything really bigger, so sugars and things like that, for sure. Again, everything we've seen is polar, so we're not going to be able to cross the membrane. So we have proteins to do this for us, and there are two overall classes of proteins that can do this, and it depends on whether or not we need energy to move the thing. So if we do not need energy, it is called passive transport, and essentially what these do, and we'll see some examples next time, is they open up a pore in the membrane or a channel, and they just sort of let it go through. And it's going to be pretty specific, so it's not just going to sort of let anything through, but so you might have a pore that opens up and lets, say, sodium ions to go through. The other option is something that requires energy, usually in the form of ATP, and this is active transport. So active transport often has a two-step process where the thing goes in, and then it may change configurations, and then it opens up and it goes out. And just whether or not a given molecule needs passive or active transport comes entirely down to whether or not that molecule wants energetically to move in the direction you're moving it. And that's going to depend on a couple things. Just another nomenclature situation here. Sometimes there are transporters that transport more than one molecule at once. And when they do this, they usually have to move both molecules. The broadest category of this is just called cotransporters. So here's a uniporter, uni for one, right, where this one substrate is going to come through. And there are two types of cotransport, one in which it moves two different molecules through in the same direction, and one in which it moves one molecule one direction, one molecule the other. Either time, though, it has to do both. So if you are a symporter that needs to move S1 and S2, there's no S2 around. You can't move S1 just by itself. Same thing with an antiporter. And this is showing these both as passive sort of channels, but just to be clear, this can work just as well with an active transporter. All right, so if passive transporters just allow movement, this is just a little bit more detail on how this works. Really, all they're doing is avoiding this situation, right? So here is our, let's say this is maybe an ion of some sort, or it could be a sugar, right? Remember, everything's always got water around it. So two things have to happen. One, I got to get it to let go of its water, and then it's going to have to pick up water on the other side. And two, you have to put it through this membrane, which we don't like to do, right? And so if you look at the energetics of diffusing without a transporter, it's very high. It might require very high energy to do that. Actually, the worst might be the getting rid of the water and then the picking up the water again. And this is probably too much energy for you to have. So what the transporter does is you still have to let go of the water, but when you let go of the water, maybe there's some nice little side chains in here that kind of replace it. So it's kind of okay. And then you've got, might have some polar sections in here, so it's okay as it's going through, and then it picks up the water again. So it just drops this. And what this hopefully should look like to you is basically an enzyme, right? We are decreasing the delta G of a transition state. So there's no chemical reaction happening, so it's not called an enzyme, but it looks a lot like it, right? If you kind of considered the substrate to be it on this side and the product to be on this side, you almost could consider it an enzyme. But again, we should not. So, but because it is similar, we can look at the kinetics very similarly, and here is our Michaelis-Menten equation we're all familiar with. And the only difference here is instead of Km, because it's not an enzyme, we've made it Kt, but Kt is identical. It's the concentration of substrate at which the speed of transport is half of the Vmax. So it's exactly what Km was. And I also note that I put the concentration of substrate on side 1 over here in both situations. So what that means is it doesn't matter for the kinetics what the concentration is on the side you're going to. It only matters on the side that you're starting at, which makes sense because if you think about that's really the substrate, right? The equivalent on the other side would almost be like the product, and we didn't see product in the Michaelis-Menten. So the only trick here is making sure you're using the right concentration, right? You can plot it in a Lineweaver-Burk plot. Same way, same thing. Right, so just one thing you easily should be able to do is calculate the kinetics of this sort of thing for passive transport. All right, then why do some things need active transport? One situation is concentration gradients. So a concentration gradient, this should be a little bit of review, but I'm possibly not really never seen it really in this context. A concentration gradient just means that when you have a semi-permeable membrane here, that if there's a different concentration of that substrate on one side than the other, you have a concentration gradient, right? So energetically, what wants to happen more is the side that has higher concentration to move to the side of lower. And what they're showing with these two arrows is that there is movement both directions, but as long as there's a concentration gradient, there's going to be more movement in the direction towards the lower concentration side than the other side. Once you hit equilibrium where the concentrations are the same, you do not have any concentration gradient anymore, and there's still movement. Some molecules are going to move from this side to this side, some are moved from that side to that side, but they're equal. So that's what makes you at equilibrium. So concentration gradients are one important aspect of why why we might need active transport. And that's because if you want to move them from the side where it's lower to the side that it's higher, you're going to be fighting that concentration gradient, right? So moving sodium from the cytosol into the extracellular fluid, you're moving against the concentration gradient, and therefore we might need active transport. There's another aspect of it, which is why I'm just saying might at this point. If it was only about concentration, you absolutely would. Whereas moving potassium from the cytosol to the extracellular fluid, because it's much higher inside than outside, that would presumably be passive. Again, if that was all that's going on. Again, there's another aspect here. The other aspect of it is these little negatives and positives over here. So this is not meant to indicate the charge on the sodium, right? Because sodium's definitely not negative. What this means is the cell itself is more negative than the extracellular fluid. And that's because there are more negatively charged things on the inside than there are the outside. And that's also going to affect it. And this is called membrane potential. And so the membrane potential is saying that let's ignore what the actual substrates are, and let's just look at, are there more positive or negative charges on each side? All right, and so it turns out that, so even if you're looking at something here where this one has a membrane potential because we have more negative charges on the left side than the right side. And so in a situation with membrane potential, negatively charged things want to move from this side to this side. That may happen even if their specific concentration is actually higher on this side. So from just the membrane potential, the negative stuff wants to move this direction. Again, everything's moving both directions, and we're talking about the overall net change. And once you do reach equilibrium, you would have equal numbers of positive and negative things on each side. Again, that may not be the same numbers of each individual substrate, a solute, really, I should say, each individual solute, but it is equal in charge. And you can measure this by literally sticking little microelectrodes into a cell on the outside of the cell. You can measure it, and it turns out that the difference is about, in most cells, it's a period, negative 0.05 volts. So that's 50 microvolts, and that's the unit they're using here. So that negative means that it's negative on the inside. So that's on average. So there are cells that are different, there are situations that are different, but that's a pretty good average that we can use most of the time. So how does this affect actual numbers? How do we know about those things and then say, okay, this is going to be passive transport? Well, we have we got math to do it, right? So the delta G of transport has to do with the concentration gradient is simply RT ln concentration of the side it's moving to over the concentration of the side it's moving from. So if we think about this, when this is higher and this is lower, then this whole term is going to be less than 1. And the natural log of things that are less than 1 is negative. So the delta G is going to be also less than, well, it's going to be less than 0. And so we know that that means it's favorable. Right? And if this C1 is lower, aka it's moving to a more concentrated side, we would have this whole term would be bigger than 1. The delta G, the natural log of something bigger than 1 is therefore positive, and now the delta G is positive, and it would be unfavorable. We know this because R is always a positive number, and this has to be in Kelvin, so T is always a positive number as well. So this deals with the concentration gradient. And for compounds that are not charged, so things like sugars, etc., this is all that matters. However, a lot of things are charged, and when they are charged, the membrane potential matters too. So if it is an ion, you have to add this is still here, and you have to add an extra term. Okay, and so for this extra term, Z is going to be the charge of the ion. So, +1, -1, +2, that sort of thing. F is just a constant that I would always give you. It's Faraday's constant. And Vm is the membrane potential. Okay, and one critical part about this is you need to, the charge on the membrane potential needs to be the sign on the side you're moving to. All right, so if you're moving into a cell, your VM should be negative 0.05 or negative whatever. If I give you a different number, use a different number. But if you're moving in, it needs to be negative. If you're moving out towards the more positive side, the way what you need to use for VM is a positive number in order for this to work out properly, right? Because what you want is for this term to be negative if you are moving in the favorable way with the membrane potential. And so that would mean that for say a positively charged ion, let's say sodium, with the membrane potential only, we would think that moving to the inside would be better, right? So we have to, and this is going to be a negative number. If we have a positive number here, F is always positive, and then a negative number. So that would make it favorable. Alternatively, if we have a negative ion like chloride, the favorable thing would be to move out. And so to make this term be negative, we need the membrane potential to be positive. So those would be favorable. And what would be unfavorable would be, for instance, positive and positive. That would lead to a positive term here and negative and negative. However, because there's this other term, when you have an ion, if one term ends up being negative and one term ends up being positive, it depends on which one is a bigger number. So if you're moving, for instance, against your concentration gradient, but with your membrane potential, you need to actually do the math to determine whether or not this is going to be overall favorable, whether or not the whole delta GT becomes negative or positive. All right, so really the best way to do this is just know this equation. And then if you know it's a neutral compound, you just cross this term out because the charge equals 0. And 0 times anything is 0. So you don't even really have to remember not to use this for something that's uncharged because it should zero out as long as you put the charge as 0. All right, so using this can be a little bit complicated. The units can get sometimes a little wonky, and this whole issue of whether the membrane potential should be positive or negative can be tricky. So we're going to do some practice problems with this in class for sure. But hopefully this was a good introduction. All right, and then the last section of Chapter 11, we're just going to go through some examples of transporters.